that we did from a pipe that was a downspout off of a house that was chewed by a purported Sasquatch. So I'm going to speak just a few minutes on the DNA portion of the study, and then we're going to pass it down to the rest of the submitters, and they're going to give you a little information, and they have a little bit of show and tell. And finally, then I brought some of the actual samples for you to look at. So hopefully this will help people to understand, and also we want to address some of the criticism that's been of the study and why. First of all, this study, we didn't plan on doing this study. I didn't believe they existed at all. When the study started, it was just some people wanting species identification. And when word spread, we ended up having a lot of different samples submitted, and it became a project. And with funding from Wally Hersom and Adrian Erickson, we were able to do extensive genetic testing on these samples. We had over 100 samples. In the press notification we sent, we have the Sasquatch Genome Project.org website where the paper is free access. You can go and read the paper and see the attachments and the photos that accompany the paper. It also gives you a detailed list of the submitters, what they submitted and where it came from. So we ask you go to those that website and have a look, because it will explain a lot of, because I can't go into the length of the paper here, we'll just address a few of the issues. We spent 5 years doing this study. We started out with just simple testing and progressed all the way to whole genomes. You've heard of the Human Genome Project. We did the same thing the Human Genome Project did, except we did it on 3 samples instead of 1. The new technology allows us to sequence efficiently and quickly. We have 3 terabytes of data from over the 3 samples, so about a terabyte of data for each sample. Then also the new technology allows us to compare it to other animals and human as far as the sequences, which we did this. University of Texas Southwestern did the sequencing. They also did the analysis. We've had other groups and other geneticists look at it. Some of them have supported us, some of them haven't. The ones that haven't, it's because there is very little alignment, meaning it doesn't match anything. It's novel, it's new, there's nothing to compare it with. Since a lot of animals and humans will share certain DNA sequences, you get a very low yield of matching data, and when you play with these genomes, you can get a large number of different animals that will show up just in 1%, 0.5%. You get human at about 3% on one sample, and this is over the whole genome, not just a portion. So we've got alignment that is not good. It doesn't fit well in the tree of life. And this is the problem that a lot of the scientists have. They expect that it's an ancient that somehow branched off with evolution. But this creature does not follow the general rule. What it does do is it's very different. We think it is a human hybrid. That is our theory. There's a couple of reasons for this. First of all, there's a type of DNA called mitochondrial DNA, and you may have seen some of the advertisements about getting your genealogy done. This is the first thing we did with these samples, and surprisingly enough, all of them tested 100% modern human. We really somewhat weren't surprised by this because 4 other laboratories have already tested these types of samples and some of the same samples and got the same results. The difference is they were not forensic laboratories, and they thought that they must have contaminated the data. What we did, we split the samples between my lab and the North Louisiana Crime Lab. They extracted the lower yield samples robotically. We extracted some of them, the large yield samples, manually, and to get the DNA to sequence it and have a look at what these creatures were. And then we ran those samples in tandem, and one of the samples in the study, we actually had enough DNA that we did 2 different extractions, and it repeated. So we got consistency there. All of them turned out human. The New York University did a number of these, and Adrian Erickson will touch on this. what happened with his sample there. It tested human. He sent it to Paleo Labs, it tested human mitochondrial DNA. He sent it to my lab, we tested human mitochondrial DNA. We have footage. This is the remarkable part. His footage of this red female Sasquatch matches her genetic profile, because he had no idea what we were going to test. And I chose to do a color gene for her hair color. And when we tested that, it turns out it was human sequence from this individual, and she had the red hair gene, and she is red in color. Also, we had only 2 or 3 females in the study, and her gender is female. And he had no way of knowing we were going to test these things. So it supports the video. And this is, very, it's good. Plus, 2 other labs prior to mine had tested this sample and got the same mitochondrial DNA to human. But we went further. We went into the nuclear DNA, which is the DNA that's inherited from your mother and daddy. And what we found there was it was not human. It had bits of human sequence in it, but it didn't align well with anything that we've ever seen. And we did a cursory look at the whole genome using just single mutations. And when we did that testing, it fell well below the human threshold. It should have been up above 95% of these mutations appearing, and it was down in the 87 percentile. And this was pristine DNA. Since we don't fit in the tree of life with these animals in a convenient way, scientists say, well, it has to be contaminated. These samples were not contaminated, because there are ways to test this. We are forensic laboratories. We deal with contamination on a daily basis. We know how to wash the samples and get rid of any contaminating DNA. If it is a liquid sample that can't be washed, when we test it with what's called universal primers, which allows the test to show up any mammal that is in the sample. And what happens if you have, say, a human and a mammal, your profiles are going to superimpose, and it will give a particular look to your graph. And when you see this, you know that it's contaminated. We had a urine sample that was contaminated. We discarded it from the study because the profiles were superimposed. We do not have that with Mr. Erickson's samples or any of them in the study. They were all pure samples. The ones that were hair were well washed prior to being extracted, so there was no contaminating DNA. And plus, when we tested it, it proved it was not contaminated. The other criticism, they said our DNA was degraded. Not only did you can put them on, you've seen these gels where you put the DNA into the gel and it runs and you see these bands. Well, the bands are going to be smeared if it's degraded. These bands were not degraded. We had in the paper the clean bands showing they're not degraded. Furthermore, we took one sample a step further. We had plenty of DNA. We had histopathology done where it was cut and put on a slide where we would see if the tissue was broken down. It was not. It was not bacterially contaminated because there was no bacteria in the slide, and we stained for bacteria and for fungi, and both were negative. So we know we have good DNA. We know it's not contaminated. So we know what we've got is the profiles we're getting on these samples, 100 and something of them, that are consistent with one another, are absolutely what they are. It's just very difficult. I call it the Galileo effect because this is very novel. It's the first time that anybody's seen anything like this. And mainstream science has a problem with that because we're all taught that evolution is very cut and dried. And this is how it is. It comes up the tree of life. But what we have is a tree of life with a single branch coming up and crossing over to human. Furthermore, we did something called electron microscopy where we expanded the DNA to the point with electron microscopy so we could see the actual structure of the DNA. And in doing so, we found that was also novel, that it would have a stretch of double-stranded DNA like you're supposed to have, but then you would have groups of single-stranded DNA interspersed in it. Even the critics of the paper have said that this is what you would see if there was a hybrid between, say, a human and something that didn't align very well with human. There's going to be gaps where it didn't fit, and where it doesn't fit, then you get these single-stranded gaps. We knew there was some kind of a structural problem when we were actually testing it. We could see it on the gels that it didn't look right. So that was why we did that one. Plus, it also supported, it had long fragments of DNA in these, in this, and if it was degraded, they would have been little bitty short. And this was repeated, and we had 2 different universities look at these results. So we've ruled out contamination, we've ruled out degradation. So the bottom line is the study is what it is, whether science likes it or not. We went to Nature first. I did not leak it, but the peer reviews have been leaked, and I encourage you to go to that link and have a look since somebody else put them out there. I'm not responsible. They had leaked a little bit of the results themselves of the peer review. And if you'll read the responses, you'll see that they didn't read the paper because they were calling it ancient DNA when it was fresh DNA. They, you know, were asking for things that were already in the paper. And there were just a number of things that were already there that should have been addressed, and they weren't. But you can read that for yourself, and it'll give you a better understanding. There was also the question about all of this is supposed to be kept confidential, but when Nature leaked, I still chose to keep it confidential, but I did make it available to the people that participated in the study, and obviously one of them let it out. We also was leaked peer reviews from a new journal called James. They were going to publish it, and then there were some legalities that came up because they were afraid of the stigma. So we ended up actually to preserve those peer reviews, and that was leaked also on his own website, where we did pass with revision, and then the editor, we corrected what they wanted, and the editor chose to publish. An hour before we were going live with the press release, we were told that they weren't going to publish because their attorney had told them not to. So we ended up, since it was a new journal, we acquired it and went ahead and published it because we tried several journals, and 3 of them would not even send it out for peer review. And they would say things, you know, like, oh, well, you don't have any proof, and not 3 genomes later, that just can't be. Or they would say, oh, that hair could have come from anything, but the hair was DNA tested. So we've had a lot of negativity from the scientific community. However, we've had other geneticists that have come out in favor. Dr. Swinson, David Swinson, he has given, you know, sterling reviews, and he went in and checked the data. We had another one that ran a genome center that did it for us, and he confirmed our findings. But his boss wouldn't let him go public with it because of the stigma. So we're fighting politics here, basically. Everybody's afraid of the subject because a lot of people don't believe it exists. We've had a lot of good feedback on the paper, but like I said, a lot of them, they're not being allowed to go public. It's very disheartening to us that worked so hard to bring this study to fruition. What we're here today to basically do is, one, to tell our side of it, because we've kept silent. We've tried to stay ethical and not, you know, voice the reviews and not, you know, get out there with what's really happened with the paper. But now Mr. Erickson decided to at least show y'all some of the footage. Some you'll get a DVD with some little short clips, but we're going to show you more than that, so you can see really good video where you can see these creatures. And like I say, the DNA supports it. I also have a video taken from my lease that's connected to some of the Texas samples on the submissions list. And I was actually there when a footprint cast was made. So you talk about chain of custody, something could have been changed out? No. I was there when we took the cast. The hair was in the cast. We pulled the hair, we put it in the study, and it was consistent with the other samples. And along that same time, we caught a video of a approximately 10-foot individual in a tree at 500 yards. And Troy Hudson he was there when it happened, and we tried to recreate it to get size, and he's going to talk about that when we show the video. So what we're doing here is we just want people to understand this is a serious study. It cost us $500,000. We have 3 whole genomes. We have a lot of supporting material of all different disciplines. One more problem is we're getting people that they'll critique one part of it, but they don't look at the whole paper. All of it comes together to make the proof. Plus the bias is there in that the Lasula monkey was recently discovered, and that I think they used 1,600 bases to identify a new species of monkey. However, we have 3 terabytes of data with billions and trillions of bases, and they won't accept ours. So, you know, because it doesn't fit neatly into the tree of life. So with that, I'm going to pass it to Adrian Erickson, and he's going to talk a little about the Erickson project and all the links he went to make sure that both the DNA and the video was pristine. Mr. Erickson.
I'm Adrian Erickson from Canada. I saw my first Sasquatch at 8 years of age in 1960. And since that time, in every decade, I've had sightings and interactions with them. And it came to the point that by 2005, there was so much evidence with the, more people coming forward on the internet that I decided to embark on this, which became an unprecedented 5-year study of the Sasquatch, but I spent a lot of money on to try to prove once and for all the existence of the Sasquatch. Dennis Poe beside me was a project manager, was on there for several years. We had an on-site scientist, and we conducted it in several states, the states in the U.S., including Texas, and 2 of the Canadian provinces. Now in that study, we collected all kinds of, we have hours of video, night vision, daylight footage, all kinds of stuff, footprints, handprints, hairs, all kinds of stuff. And part of it was we followed a mother and her juvenile daughter filmed them over a 4-year period, of which the daughter grew approximately a little less than 6 inches per year. And it became, with all this evidence, it just seemed like the scientists would just raise the bar higher all the time. And finally it came to the point that we just said DNA was the only thing that would suffice to convince them. But with no type specimen, DNA is all fine, and on CSI everybody thinks it does all these things. You have no type specimen with a Sasquatch, so we knew it wasn't going to be easy. So anyways, with us, during that project, we collected 6 different samples that was tested by Dr. Melba Ketchum, and Dennis was mostly the guy in the chain of custody and control of that. Now also during that time we produced a documentary, a 1-hour documentary that I took to several producers, and at the end of the day they liked it, but the whole reality world, that was, reality television, that was the key thing, and that's what they wanted us to do, and I wanted no part of this. I mean, this is serious business. I mean, at an absolute minimum, there's 4,000 of these in North America. In reality, it's becoming increasingly apparent there's 3 to 5 times that number, or even more. And yet, people have chosen to they don't want to believe it. They can't find in their minds to believe these things exist. Now, their survival skills and intelligence are far beyond ours. I mean, they don't have to cook their food to get all the nutrients out of it. They don't need fire for warmth. I mean, they have incredible senses, especially their exceptional night vision. I've often said, if I were covered in hair, if I didn't require fire to cook my food or provide heat for myself, I had the night vision and the smell and the intelligence that they have, and I knew if I exposed myself to one human that I might be shot, nobody would ever see me. They know how to cover their tracks. They have a sixth sense we simply don't possess. But many wild animals have that sense. A track is a signature of a living species. There have been thousands of tracks cast. There's thousands of tracks all over the world for that matter, and yet nobody takes it seriously. I'm a bow hunter. I've made 37 safaris to Africa. Most of our game is hunted by tracking. Some of those animals you'll never see, but you know they exist because they leave tracks. And yet, again, the scientific community at whole simply ignore that. The huge stigma with this creature, in fact, the greatest detriment to its recognition are the scientists themselves. To me, personally, the biggest problem, I think. I mean, governments of both Canada and the United States, we know they know. I mean, the RCMP, which is the Royal Canadian Mounted Police of Canada, and the Fish and Wildlife Service have, off record, have admitted to me that they certainly know they exist, but they've been told to deny and diffuse. Now, part of it is the, the economic fallout that could happen in this country could be huge. Look at the impact the northern spotted owl had in the Pacific Northwest. Sasquatch have been reported in all 49 continental states and all 9 continental provinces in Canada. Now, back to the scientists. It's just, scientists like to be safe, they rely on funding, they don't want to make a mistake, but it's very easy for them to criticize the efforts of lay people because they haven't put any money into it. I mean, there's been hundreds of millions of dollars spent on the overstudy of the grizzly bear or marine mammals. Not one penny has been spent on funding Sasquatch research. It's left to lay people. And it's just easier for them to criticize the work of us. I mean, in the words immortalized by your late President Theodore Roosevelt, it is not the critic who counts. Not the man who points out how the strong man stumbled or how the doer of deeds could have done them better. The credit belongs to the man who's actually in the arena, whose face is marred with dust and sweat and blood, who strives violently but errs and comes short again, who knows the great enthusiasms, the great devotions, and spends himself in a worthy cause. And at the end, at his best, will know the triumph of high achievement. But at the worst, if he fails, the least fails while daring greatly, so his place shall never be with those cold and timid souls who know neither victory nor defeat. This has been the most frustrating experience in my life. I didn't set out to make money. I set out to recognize the species and to vindicate the tens of thousands of people who have been severely impacted in their lives by simply coming out and telling the truth. Now, truth goes through 3 stages. The first stage, absolutely ridiculed. The second stage, violently opposed. And the third stage, it's accepted as self-evident. We don't have a mountain of evidence, we have a mountain range of evidence. Every other species recognized on this planet has been done so with less than 1% of the evidence the community at whole has. I mean, I've got hours of footage. I've got hundreds of hours of interviews with some of the scientists that came to our sites that actually saw Sasquatch and all this stuff. When somebody takes it seriously, I will present a bunch of that footage. But you know what? Eventually, the Sasquatch will be accepted. Thank you. I'll turn it over to the project manager, Dennis Pope.
Before Dennis starts, I'd like to point out that just about everybody at this table has seen them, has had an experience with them. We come from both a field and a scientific background. I didn't believe them, and I was able to go out and 3 years ago and start having experiences, and I've seen them multiple times since. Most of us have seen them multiple times and had multiple interactions. And Dennis has huge amounts of experience with them because he was in the field. So Dennis, would you like to go from there?
My name is Dennis Foll. I was involved with the Erickson project. My name is Dennis Foll. I was involved with the Erickson project. Became involved in September of 2005. Adrian and I were part of the Bigfoot Research Organization, and that's how we met. And basically, I committed to 5 years of trying to keep this project organized and work with individuals at the location in order to gather substantial evidence on the existence of the Sasquatch. This particular area had what I would call repeat visitations by a small family group, and we were able to gather some interesting bits of video evidence as well as several casts and audio recordings. So it has definitely been a very interesting 5 years, and I would do it all over again if I could. Yeah, that's basically my role in this whole thing. We did, as we went along in this project, we tried to improve in our processes to gather evidence, which was very difficult. The video evidence was difficult because we're working in an environment that is difficult to film in, for instance, usually after dark and thick woods, and you had to have specialized equipment, night vision devices, thermal imaging. Adrian thankfully provided us with the funding to purchase the necessary equipment to do this, and that did help tremendously, and as well as consistency, being out there and constantly working to gather evidence. That was that took thousands of man hours. I can't even begin to calculate the efforts it took to gather what little we do have, but it was quite an effort, and it's something that does not come easy. And even with the situation we were in, it was a difficult process to do. And we had a family group that was pretty well confined to this area that we were able to get close to from time to time. So that did help us in the entire project to get what we do have. So basically, we have compiled this together. We've got video, we've got several casts and hair samples, and eventually we decided that the necessary steps was to gather DNA. We were told by several prominent scientists in the field that would be the next step, the next logical step. And so we took on that effort, and were successful eventually. And that DNA was submitted to Melba, and Melba basically explained that earlier. Other people have contributed to that effort, and we're pleased with that, but apparently that still is something that is not an open and shut case for certain scientists in this field. So other than that, I really don't have anything else to add to it. So
you might add that you collected one of the genome samples.
yes, yeah, I did say we were successful in collecting, and I do have a photograph of one of the samples that we collected that Melba has processed, and I'll show that here in a little bit.
Okay. Next is Rich Brumeau from Washington State. He is co-founder of the Olympic Project.
Yeah, my name is Richard Germeau, spelled G-E-R-M-E-A-U. For some context, I was in law enforcement for 14 and a half years. In the year 2000, I was driving into work in my patrol car, and one of these things stepped out in the road in front of me and took 4 steps and crossed. I hit my brakes. I was about 50 yards from it. Before that, I didn't believe in any existence, and it was kind of a traumatic event, even though I was inside my car, just because I was attempting to classify it. And that's what I did in my job, was to observe and to classify, and I couldn't put it in any box where it belonged, even though I knew where it was. But anyways, moved 10 years forward, and I ended up getting in contact with a guy who seeked me out to get involved in this research because of my background, and I was credible. I was a major crimes detective at that time, and I was on the SWAT team and whatnot. I did a lot of forensic evidence